Hi, I'm Yayu. Today I'm going to talk a little bit about the role of glucocorticoid in regulating the function of microglia. So my first year project is focusing on the stress and its related hormones, glucocorticoid. So I chose this paper to present. It is well known that psychological stress-induced elevation of glucocorticoids can suppress immune responses. We can see this inhibition in the central nervous system, especially in microglia. Previous studies have shown that glucocorticoids are able to directly inhibit microglia function and suggest a possible role for glucocorticoids in the regulation of microglia function in vitro. However, there is also in vivo evidence suggesting that glucocorticoid and stress actually can enhance immune function within the central nervous system. Therefore, determining how stress affects microglia function is essential for understanding glucocorticoid-induced central nervous system inflammation. In addition to change the amount of inflammatory mediators, glucocorticoid also raise the excess release of glutamate, induce NMDA receptors overactivation, and cause neuronal death within the hippocampus. Therefore, glucocorticoid-induced NMDA receptor activation within neurons may provide a stimulus for microglia activation during periods of stress. Previous studies have shown that glucocorticoid increases the susceptibility of the brain to a variety of insults, including hypoxia, ischemia, and excitotoxic lesions, are not only attributed to neuronal loss, but can also activate microglia. This excessive activation of microglia leads to destructive central nervous system inflammation. Therefore, determining how stress affects microglia function is essential for understanding glucocorticoid-induced central nervous system inflammation. So, in this study, the author used the male C57BL6 mice to study this topic. Mice were physically restrained in a tube. This procedure induces psychological stress because of the animal's confinement. Mice were restrained for 50 hours a day for a total of 6 days. On each of these days, a subset of mice was sacrificed and brains were removed to analyze for flow cytometry, standing for microglia for pCNA expression or annexin 5, serine collection, and corticosteroid radioimmunoassay and immunohistochemistry. Next, to determine whether glucocorticoids affect the function of microglia, the author used the restraint stress model to elevate serine corticosteroid levels. On each of these days, microglia of mice brain were quantified by flow cytometry following staining with anti-CD11B and anti-CD45 antibodies. The results show that microglia numbers are increased during restraint stress. Then, to verify the effectiveness of this restraint stress model in elevating corticosteroid, mice were examined for serine corticosteroid levels in these two groups. As expected, the restraint stress produced significant increases in serine corticosteroid. This indicates that the increases in serine corticosteroid observed in the stress group were indeed the result of restraint. Next, the author tried to examine whether 4 sections of stress was minimally required to cause stress-induced microglia increase. As seen in the graph, only following 4 sections of stress was a significant increase in microglia numbers from baseline observed. This finding indicates that the increase in microglia number occurs specifically in response to 4 sections of stress. So, we already know that the stress increases microglia number. It was still unclear whether these increases were due to microglia proliferation. So, to determine whether microglia proliferation was occurring following restraint stress. We examine microglia from stress mice for their expression of proliferation cell nuclear antigen (pCNA). The results show that the stress induced both pCNA expression and apoptosis in microglia. This result verified that increases proliferation observed on day 6 is offset by an increase in apoptosis on the same day, preventing any significant increase in microglia number on that day. Then, the author proposed a mechanism that stress-induced elevation in glucocorticoids begin a cascade of events leading to microglia proliferation. The glucocorticoid level will increase after restraint stress. When glucocorticoid receptors are stimulated, increases in neuronal activity and decrease in glutamate uptake leading to accumulation of extracellular glutamate. This triggers activation of NMDA receptors. One consequence of NMDA receptor activation is the production of various pro-inflammatory mediators. These mediators may be responsible for stress-induced microglia proliferation. To determine whether this pathway is involved in stress-induced microglia proliferation, metilrepone, RU486, and MKA01 were used to block the glucocorticoid receptor and NMDA receptor. These findings show that the increases in microglia is associated with stress are mediated by the elevation of corticosteroid levels. Next, to verify the effect of metilrepone, serum corticosteroid levels were measured 3 hours into the stress section. Restraint stress increased serum corticosteroid level on each day of stress. Metilrepone only blocks stress-induced elevation in corticosteroid for the first 2 stress sections. These findings suggest that blockade of the corticosteroid during the first 2 sections of stress is sufficient to prevent stress-induced proliferation of microglia on day 4. With above findings, the author hypothesized that the treatment of mice with exogenous corticosteroid-induced microglia proliferation. The results clearly indicate that exogenous glucocorticoid administration increases microglia numbers. So we have known that stress induces high corticosteroid levels and binds to the glucocorticoid receptor. Therefore, binding of the glucocorticoid receptor by corticosteroid may play a role in observed stress-induced increases in microglia. The results show that blockade of glucocorticoid receptor can prevent stress-induced increases in microglia. This suggests that corticosteroid and glucocorticoid receptor binding is required for microglia proliferation in the stress model. Then, the author also found that blockade of the NMDA receptor within the antagonist MKA01 would be expected to attenuate stress-induced microglia proliferation, and the results indicate that the corticosteroid-induced increases in microglia numbers are mediated by activation of the NMDA receptors. Finally, due to the previous studies show that the hippocampus is especially vulnerable to the toxic effect of stress, the author examined stress-induced elevation in microglia numbers within the hippocampus. The results show that the treatment with the metilrepone, MKL1, or RU486 can prevent stress-induced increases in the number of activation microglia macrophage within the hippocampus, and neither of these groups was significantly different from baseline. This indicates that stress increases the number of activated microglia within the hippocampus through glucocorticoid or corticosteroid-induced NMDA receptor activation. Okay, finally, there are 2 take-home messages. First, these findings demonstrate the elevation of glucocorticoids are able to activate microglia in vivo, and suggests that stress is able to induce a pro-inflammatory responses within the central nervous system. A pro-inflammatory microglia response may be a contributing factor in the development of various stress-induced inflammatory conditions in the central nervous system. Second, future studies should investigate the mechanism of the elevation of corticosteroid level and NMDA receptor activation stimulate microglia proliferation. Also, researchers should focus on whether neuronal-derived inflammatory mediators are responsible for this microglia proliferation. That's all I have today. Thank you.